is an overview of the Biomarker Services cellular phenotypic platform from Eurofins Discovery. The Eurofins Discovery Biomarker Services platform provides a flexible analytical approach to evaluate a wide range of different biomarkers in a number of different systems as best suit the particular project or study needs. In a very general sense, we are able to evaluate a range of different test articles of different types and apply them to a variety of different cellular systems, for example PBMCs, and then measure the effect of those test articles in the form of a range of different biomarkers. This may include soluble biomarkers or secreted biomarkers, such as cytokines, but can also include biomarkers that either are cell surface expressed or intracellular biomarkers. In addition to the evaluation of biomarkers in relation to cellular systems, we are also able to evaluate biomarkers in a range of different biological samples. For example, samples taken from in vivo studies where biomarkers of interest might be wanted to be evaluated in samples such as serum, plasma, different tissues, and extracts. We can thus evaluate biomarkers in a range of different environments as best suit the needs of a particular study, and we have at our disposal a range of different technologies that will allow us to do so. As far as cell types go, we have a bank of peripheral blood mononuclear cells, or PBMCs, available as cryopreserved banks, and this covers a range of different human subjects with a variety of different ethnicities, ages, and both male and female. We use the PBMCs for several of our Catalogue assays, but we are also able to use the PBMCs in a variety of customized assays, again, as best suit the needs of the particular study or program desired. In addition to PBMCs, we can conduct studies in a wide range of different cell lines and cell types to meet a diverse variety of different needs. For example, we can do studies in whole blood or fractions of whole blood. We can also conduct studies in a range of different established tumor cell lines, for example, JERCAT or THP1 cells. And in fact, we can actually access and use all of the cell lines in the Eurofins Discovery OnCope panel platform. In addition to tumor cell lines, we can also conduct studies in a variety of other primary cell types, and this includes cells such as HUVEC cells. And in fact, we have an assay available to evaluate the effect of test agents on vascular inflammation in HUVEC cells. In addition to conducting studies in human cell lines or cell types, we can also conduct studies in cells from different species. For example, we have PBMCs available for use that are from mouse, rat, dog, pig, and non-human primate origin. And in fact, we can conduct studies in a variety of different cell types and cell lines from any of these species as they are available for use. We can also customize the cell type to be used in a particular study, and this might include engineered cell lines, or perhaps cell lines that are available for provision to us for use in assays by the client. Obviously, in such situations where a client is providing a cell line, and that may be proprietary to the client and not commercially available, we would only use those cells in studies that are authorized and directed by that client. As far as technologies go, we have a wide range of different platforms available, and we select the best technology to use for any specific study based on its application. For a large number of our Catalogue assays that include assays to evaluate cytokine release from PBMCs, we use the Luminex platform. This allows us to provide multiplexed readouts from the same set of treated cells, thus providing efficiency and economy on both use of test samples and test compounds, as well as the evaluation of different biomarkers from the same set of treated cells. In addition to the Luminex platform, we have available the mesoscale discovery platform, and we conduct a large number of ELISAs routinely as part of our day-to-day operation in the Biomarker Services platform. Using these technologies, we can evaluate a wide range and diversity of different biomarkers that are either secreted proteins, for example cytokines or growth factors, as well as biomarkers from protein extracts, cell or tissue lysates. We can also use HTRF assays or homogeneous time-resolved fluorescence assays to evaluate biomarkers as are suited to that particular platform. And we have recently added flow cytometry to our capabilities as a new technology that allows us to evaluate not only cell surface biomarkers, for example cell surface antigens, but also a range of intracellular biomarkers. And furthermore, we can even evaluate biomarkers on a cell type basis. For example, in a PBMC population, looking at a variety of cellular biomarkers in one PBMC cell subtype as compared with another. We also have at our disposal a high-content imaging system, and we can employ this to look at biomarkers on a more close inspection basis, where we might be, for example, looking at the nuclear translocation of a biomarker, or other biomarkers that might be better suited to evaluation in fixed cells by high-content imaging. Lastly, and by no means least, we have extensive mass spectrometry capabilities available, and we can use those either in parallel or instead of some of the above technologies for the evaluation of biomarkers where there are either not commercially available reagents to detect those biomarkers, or the biomarkers themselves may not be directly amenable to detection using the fundamental technologies in the above platforms. As I'd mentioned, we'd recently added flow cytometry services and capabilities to our Biomarker Services platform. And with this, we are able to conduct a wide range of different evaluations. Some of the examples of this are shown on this particular slide. This includes things like immunophenotyping. As I'd mentioned, we can often evaluate the effect of a particular test agent on different cell types in a mixed cell population. We can also use this platform to evaluate receptor saturation, for example, using the ABCB technology. And then reading down the list here, there are a wide variety of different biologies and different biomarker types and in different contexts that we can evaluate using the flow cytometry platform. We currently have 12 colors available, so we are able to also multiplex different readouts using this platform in addition to more simple single readouts. As far as catalog assays go, we have a number of different assays already established and available for use, and these are largely directed towards the evaluation of test agents in an immunological context. This includes a cytokine storm assay that can be used more from a safety testing perspective, and we'll talk about that in a second. It also includes a range of different assays that are directed towards evaluating test agents for their effect on immune cell function. As a subset of those assays, we also have a suite of toll-like receptor assays, wherein we can evaluate test agents for their ability to modulate toll-like receptors. And both the immune cell function assays and the toll-like receptor assays can be used either in a direct therapeutic activity perspective, looking for either activators or inhibitors of the particular system, or in an inhibitory perspective, where one is looking for inhibitors of those particular targets or cell types. We do also have other assays available. As I'd mentioned earlier, we have an assay to look at vascular inflammation, and we also have an assay available to look at the STING pathway. For the cytokine release syndrome or cytokine storm assay, we employ, as I discussed previously, one of our primary technologies, which is the Luminex platform, to evaluate the secretion of a wide range of different relevant cytokines that are indicative of a cytokine storm-like response. This is a particularly useful assay because it can be applied to the development of a range of biologics and even certain agents that are designed to improve the immune system to make sure that an unwanted and uncontrolled pro-inflammatory response, or the cytokine storm, is not elicited from PBMCs as a consequence of their exposure to those test agents. The list of cytokines shown here on this slide comprises a number of different pro-inflammatory cytokines that have been recommended by regulatory agencies for evaluation in such assays. And in doing these assays, we can then choose, or we can work with our clients to choose a range of different PBMCs, either cryopreserved PBMCs that we have in our bank, or using fresh PBMCs that are available from our vendors. We can use this particular assay in customized format, and of recent times there has been interest in using this assay in an inhibition mode, wherein we can actually look at the efficacy of different test agents for their ability to inhibit a cytokine storm-like response. And this is particularly applicable with the advent of the COVID-19 situation in 2020. We can customize the assays, again, using PBMCs of non-human origin. This can include studies that are designed as preludes to in vivo studies, for example, using mouse PBMCs or rat PBMCs, or as a prelude to tox studies, for example, using PBMCs from non-human primate, dog, or pig origin. In terms of presentation of the test agents, we can present them in a variety of different formats, including soluble presentation, which mimics the effect of the agent in free circulation. We can bind the test agent to the plates, and this is most applicable for biologics, by wet-bound or air-dried presentation. In both of those cases, we have the potential to evaluate other epitopes, especially within biologics, that may be exposed upon binding of the biologic either to its target or clustering, and thus expose epitopes that might potentially cause a cytokine storm-like response. We have validated assays for the cytokine storm assay for exposure times of 24 or 48 hours. And we can also provide, as a parallel study, an evaluation of the effect of test agents on proliferation or viability of the PBMCs being used. This can be accomplished using standard BRDU ELISAs or the Alamar Blue viability assay. And the benefit of doing this allows one to evaluate the correlation, if any, between modulation of different cytokines released with the number of PBMCs that are present after the exposure to the test agent. We have a range of other immune cell modulation assays available, including assays to evaluate the effect of test agents on T cells. And all of these assays, and as I'd mentioned, including the cytokine storm assay, can be set up in both inhibition and activation format. Simply put, in an activation format, we will evaluate the effect of the particular test agents on the cell type being used, largely PBMCs. And in those activation assays, we are comparing the effect of the test agent with known activators either of the cell type or the TLR being examined. When set up in inhibition format, we will actually use those specific stimuli or ligands as the primary stimulus for the cell type and the biological system being studied, and we will pre-incubate with the test agent before stimulating to evaluate the effect of those test agents in an inhibitory capacity. In addition to the T-cell functional panels, wherein we have assays to evaluate activation or inhibition of T cells when stimulated by a range of different agents, including classical T cell activation agents such as anti-CD3, but also mitogens and other agents such as CON-A and so forth. For each of those stimuli, we have a panel of cytokines that is indicative of the particular mode of activation of the T cells. And similarly, we have assays available to evaluate the effect of test agents on macrophage and monocyte function. Again, we have a panel of cytokines that are indicative of the function of that particular cell type, wherein we are using LPS as a comparative activator, or LPS as the stimulant in the case of inhibition assays. We have a range of toll-like receptor assays available, specifically targeting toll-like receptors 1 and 2, 2 and 6, 4, 5, 7 and 8, and 9. And for each of those assays, we have a panel of cytokines that are indicative of the activation of those specific toll-like receptors. Again, in activation mode, we are using the ligand that is specific for each TLR as the positive control for comparison with the test agents. And in inhibition mode, we are using those ligands to stimulate the cells via the specific TLR, and we are looking there to see if the introduction of a test agent is capable of inhibiting that ligand-specific stimulation. The Biomarker Services platform is a very highly customizable platform. As I had mentioned occasionally throughout this presentation, all of the assays that we have can be customized as best suit the needs of the client or the specific program for which the assay is being run. We have the ability, for example, to alter stimuli that are used in some of the assays, the cell type that is being used, the incubation time that is being used, the biomarkers that are being measured at the end of the assay, or, for example, we can actually develop assays from the ground up as best suit the needs of the particular program in question. In the case of customization, and especially in the case of development of new assays, we are able to use any commercially available kits or reagents in order to be able to do so, providing that those agents are not only available but free from restriction of use. But also, we are able to use reagents, for example, cell types or other reagents of a more specific nature that can be provided by the client. As I'd mentioned previously with respect to cell lines provided by a client, in cases where that is used, we would only use those reagents or cells specifically for that client's study, after which, the completion of which, we would destroy or dispose of the reagents, unless the client wanted us to keep them for subsequent assays. In all cases where we are developing customized assays, we can do this, and we actively do this in collaboration with the client to design an assay that best meets their needs and provides meaningful and robust data. We have also, as a measure of the customization of the platform, expanded the service to accommodate many of these new needs. This includes the addition recently of new PBMC donor samples to our bank of cryopreserved PBMCs. And for those, we have phenotypic data on the PBMC samples, allowing specific selection of PBMCs from certain donors over others. We've also expanded the technology capabilities to allow new possibilities, and that is best demonstrated by the inclusion of flow cytometry as a technology platform within Biomarker Services that now allows us to conduct a wide range of additional studies that expand our ability to evaluate biomarkers from outside the cell to the surface of the cell and on into the cell. So collectively, with the range of technologies, approaches, scientific expertise, and know-how that we have available, we are able to either execute or design and execute assays to evaluate biomarkers across the whole panoply of different biologies that one might like to study. The Biomarker Services platform offered by Eurofins Discovery is thus a very innovative, flexible, and very high-quality platform. We pride ourselves on the quality of data that we provide, and it provides a very powerful, informative tool to measure and evaluate a wide range of different biomarkers in a wide range of different biological systems. We have a variety of technological innovations that we've employed, including expansion of technologies. I've mentioned obviously the flow cytometry capability that we've included, but also movement from 96-well assays into 384-well format, thus improving the throughput and in some cases economy of samples that are used for particular studies. We have a range of new assays, including things like the STING pathway assay, again, expanding our capabilities into the realms of other areas of biology, again, with a focus on looking at particular biomarkers relevant to those biologies. We've employed cutting-edge evaluation of biomarkers in a translational phenotypic model systems. This includes not only the biomarker services that I've outlined in this presentation, but also some of our other phenotypic services platforms, for example, the Biomap platform, for which we can provide more information there, wherein mixed co-culture systems are used to evaluate a wide range of different biomarkers in highly translational phenotypic systems. As I've mentioned, we have tremendous project flexibility. This can thus allow the tailoring of either existing assays or catalog assays to meet a particular project's needs, or we can develop assays either from the ground up or using protocols that a client might be providing or following protocols outlined in the literature. We can employ a wide variety of different technologies to use, and we can either use them on a single technology basis for relatively simple studies, or we can combine different technologies as best suit the needs of that particular study. We have the opportunity to select desired conditions for the assay, and that allows us to evaluate the effects of test agents on different biomarkers using conditions that are of the greatest relevance to the particular client's project or program. As I'd mentioned, we can develop assays de novo from the ground up, and again, using available commercial reagents and resources that best suit the particular project's needs. As I'd mentioned, this may include use of reagents that are available only from the client, but we will work with the client to develop or to put in place assays that provide the greatest value for them, and most importantly, provide the most robust, meaningful data to help drive their drug discovery programs forward. With that, I'd like to thank you. If you would like to receive more information about this platform, please reach out either to myself or to Eurofins Discovery Phenotypic Services. We would be more than happy to provide information on any of our platforms, our capabilities, or discuss specific needs on a program-by-program basis. Thank you.